Anyway, good morning, everybody. So I represent here the research part of the community, and we all witnessed explosive growth of this field with all of the advances, opportunities, but also hype. And so as we kind of study this research question, we need to ask like very fundamental questions in the aging science. And apparently there is no agreement on some of the questions like this. So, and the question is, the first question that we need to ask, what is aging if we study this process? And apparently there is no agreement in the field because some of us think of this process as increased chance of dying, and some other people think of loss of function, and some other people think, for example, as continuation of development or damage accumulation. And it's important to ask this question because depending on the answer, we would develop completely different strategies. We need to ask ourselves whether we work on, for example, decreasing chance of dying, or we want fewer diseases, or we want to preserve our function. So my view on this is that aging begins on the left with the accumulation of molecular damage. And eventually, this results in functional decline, in incidence of diseases, and eventually mortality. So we could operate at various levels of this kind of flow, but ultimately, I think it would be important to address the true kind of concept of aging. How can we actually target the aging process itself? This is not easy, and there are, I would say, not so much research in this field as of now. So, to begin address that question, we developed a research program. So we work in several directions. On the left, you can see this part of longevity. So we here address the question of the potential of the system to live long. These are not age-related changes. This is something that would make a system long-lived. And we study this at the level of cell types. For example, we could have cells which are short-lived, like cells in the blood, or long-lived, like cardiomyocytes or neurons. And we ask a question, what does it make long-lived cells long-lived? Another type is we study longevity across mammals, because some mammals, they're short-lived, like shrews. They only live 2 years. And some are exceptionally long-lived, like whales. Some of the whales can live more than 200 years. So over evolutionary timescales, nature is able to adjust lifespan over 100-fold. And how does it do it? So we try to understand molecular mechanisms of these processes. And the third one is longevity across interventions. And here we already know that some interventions in model organisms are able to extend lifespan, like calorie restriction or rapamycin, or, for example, deficiency in growth hormone receptor. So how do these processes lead to lifespan extension? And whether these processes are the same across interventions, across mammals, and across cell types. At the same time, we also look at age-related changes, which is something on the right here. So these are patterns which change with age as we grow older. And this can be quantified by so-called biomarkers of aging. So clocks, for example, epigenetic clocks, transcriptomic clocks, also causal biomarkers. They result in actionable kind of biomarkers of aging that can be used to quantify the aging process. And the third component in the middle is rejuvenation. This is something very new in our field. This is a question whether we would be able to actually reduce the biological age of a system, whether it would be some cells in the organism, or maybe a tissue, or maybe the whole organism. And the reason I placed it separately from the aging part is that the change from the younger to older state is not necessarily the same. It's not necessarily opposite, I would say, from the older to younger state. For this, we need to address that questions of rejuvenation separately and develop its own rejuvenation signatures. And altogether, it allows us to target the aging process, try to kind of slow down and also reverse, at least in part, this process. So we begin by studying long-lived species. So over the years, we studied some of these and many other species that achieved exceptional longevity. We sequenced the genome of these organisms, analyzed various mechanisms. And what we find is that very often the particular processes, genes and pathways which lead to longevity, they are quite different. For example, bowhead whale developed certain processes, but naked mole rat, which is on the right here, it has some other adaptations. And on the left here, you see also another exceptional organism called Brand's bat. It's one of the smallest mammals, but lives more than 40 years. But more recently, we try to understand this at the level of the entire group of mammals. And in the most recent research, which was published about a month ago in Cell, we analyzed 41 species of mammals at the level of multiple organs, liver, brain, and kidney, and to some degree 3 other tissues, to identify what we call longevity signatures, which is also the title of my talk. These are omics-based patterns which indicate, illustrate, I would say, longevity, the potential to live long across mammals. And some examples of here shown here on the slide. There are 3 genes, just representative examples that just to show how it works. And 3 genes and then 3 organs. You kind of see them changing with age, not with age, but with the increase in lifespan. And what is interesting is that many of these genes that we observe, if they increase with the increase in lifespan in the liver, they seem to be also increasing in the kidney and also in the brain. There is some consistency across tissues. This is quite unexpected. But regardless, we identified about 500 to 1,000 genes in each organ that represent these so-called longevity signatures, which is again the potential to live long. Separately from that, we identify these patterns across known interventions. And this is Spearman correlation matrix, which is shown here. And on the left, you can see a list of interventions which are already known to extend lifespan in mice. Like, for example, CR is calorie restriction, MR is methionine restriction, and you can see like FGF21 overexpression and so on. The red kind of cluster on the top left indicates that many of these interventions, they change the system in very similar way. Whether we, for example, subject an animal to calorie restriction, or like remove growth hormone receptor, or, for example, overexpress FGF21, mice kind of are modified in very similar way, mechanistically. On the other hand, there are several interventions shown on the lower part. For example, rapamycin, or protandim, or c-MYC deficiency, which also lead to lifespan extension, but modify the system in different ways. Apparently, and it's clear from many examples now, is that we have multiple kind of routes to longevity. We could modify the system in various ways so that it is longer lived. Although many of the changes that we've so far observed as a field, they are quite consistent. For example, this calorie restriction, methionine restriction kind of cluster is very dominant. Now, we want to relate what we observe across species with what we observed across known interventions and what happens during the aging process. And this is a very new findings, which is like, I think, new realization, which is, I think, quite important. Basically, what you see on the upper left is the cluster of longevity across species. The omics patterns that I described to you, across the liver in mammals, kidney, brain, and so on. They are all related, they cluster with one another. So what we observe, again, in the brain, kind of similar to what we observe in the kidney as we discuss longevity across species. The cluster on the lower part, which is in green, is longevity across interventions, calorie restriction, growth hormone, and so on. And they also cluster with one another, as I already mentioned, many of them. And the cluster in the middle in red, these are age-related changes. We find age-related changes at the omics level, for example, in the kidney, in the liver again, or across mammals, or across animals, and so on. And they also cluster to one another. But now, when we relate 3 different types of kind of age-related or longevity patterns, it's quite surprising that what we find is that longevity across species blue, does not correlate with longevity across interventions. So apparently, what we find in the lab, at least so far, calorie restriction, methionine restriction, growth hormone deficiency, and so on, even rapamycin, is completely different from how nature extends lifespan. And nature is able to expand lifespan 100-fold. And I think this whole potential is completely untapped so far. That's very interesting. Another interesting feature is that there's a mild correlation between longevity across species and aging. In a way, the way nature modifies mammals as they live longer is to make them older, not younger, which is quite surprising too. The way we think about it, which is still a hypothesis, is that during the aging process, we not only accumulate molecular damage, but organism also responds to its molecular damage, activating protective responses. We call them protective or adaptive responses. And these same responses, they are also observed when we analyze mammals. And therefore, presumably, this correlation reflects simply that the long-lived species are better protected. So now we can relate what we observe here, patterns of longevity across species, which stands for ML, which is maximal lifespan, and AW here in blue is adult weight, because longer-lived species, they also tend to be larger, like whales and so on. So, because they correlate with one another. And then we relate this to aging, which is in red, like circle in red, and also in green circle, which is interventions. And what we find the processes which are related to this mechanistically. And these are some of the key processes, molecular processes that we find that are related, and you can see how they're related. In red, it's a positive correlation, blue is negative correlation. We find, for example, ribosome, which is a major cellular process, is highly related to longevity and aging. We find mitochondrial, for example, translation as well, or oxidative phosphorylation, and other processes that you see here. Of course, this is just the beginning. We don't fully understand the basis of longevity, whether we talk about interventions or whether we talk about mammals, but I think this provides a first glimpse of the mechanistic understanding of these processes. Another important feature of these longevity signatures is that they provide a direct tool to identify new interventions that may lead to lifespan extension. And because these are omics patterns, we could simply screen public databases. Whoever somebody has done RNA-seq and submitted to database, we could screen them with these signatures and ask a question whether this particular modification or treatment has a potential to live to longer life. For example, on the top part, you can see like first row in red is hypoxia. Hypoxia is a good candidate, we think, that might lead to lifespan extension, even though it has never been tested for this effect. Mild hypoxia. It is related to many of the signatures that are shown on the bottom. And then the part called CMAP, lower part. We've done chemical screen with our signatures to identify new interventions. And these 4 interventions are shown in here, like, for example, KU compound, ascorbyl palmitate, AZD, rilmidine, and so on. And so some of them we already tested. For example, we published about a month ago that actually this KU compound, which we predicted several years ago, indeed extends lifespan. There was another paper just published in collaboration with other researchers on rilmidine, which we also predicted recently, and apparently it also leads to lifespan extension. And other compounds are still being tested, and we have many other compounds, and we have actually currently a very large kind of study where we test many of our predicted interventions. And so far success rate is about 50%, which tells that these longevity signatures indeed are very useful in identifying completely new ways to extend lifespan. Now I'd like to switch a little bit to what we call aging signatures. I was telling you about longevity signatures, which is, again, which indicate the potential to live long. But now we look at age-related changes. And this is something which could represented by molecular processes that change with age, and also very well represented by so-called biomarkers of aging, or clocks, which is the concept which is pioneered by Steve Horvath. And here is, I will just highlight a couple of recent studies, because now it's a very large field. There are many studies, and of course, many labs contribute. I will just show you a couple of recent examples from my lab. In this study, which was published 3 months ago in Cell Metabolism, we found that biological age, in contrast to chronological age, does not steadily increase with age, but fluctuates. So it's apparently under severe stress, biological age can rapidly increase, and once the stress is relieved, the biological age goes down and can go back to normal state. And we described 4 examples of such severe stress, which is like a hip fracture, for example, leading to emergency surgery. Also severe COVID, when somebody gets to ICU and ventilator, biological age increased, and then once the person recovers, it's decreased. Also pregnancy. Apparently, we find that women in the 3rd trimester have higher biological age, and postpartum, this age decreases. And we also found that this example of parabiosis, when we connect mice so that they have a joint circulation, and in this case, younger mice, which are connected to the older mice, they rapidly become older. But once we disconnect the mice, they recover and become younger again. So these are 4 examples that we described. Again, so this is only the severe stress. Mild stress doesn't do it. And another example is actually also on parabiosis. And this is actually the example which I think is illustrative of rejuvenation. Here also it's a collaboration with Jim White at Duke University and some other researchers, including Steve Horvath and several others, and was done by Bohan Zeng, who he's a member of my lab. We connect mice for 3 months. And then after 3 months, connect mice, young and old mice. After 3 months, we separate them and follow for another 2 months. And then later, we also follow them until they die. What we find is that old mice, after they are connected to the young mice, they are longer lived. So we just are able to extend lifespan of these mice by simply, when they have joint circulation with younger mice. And also based on the clocks, this is on the right part, we analyze several tissues. The tissues also become younger. In fact, the effect is very strong initially, and it persists after 2 months. After we separate the mice, 2 months later, the old mice are still younger, and eventually they are longer lived. Although this lifespan extension, as you can see, is not huge. So it seems like the effect is stronger initially, and over time it diminishes, but still a little bit remains so that they are longer lived. So maybe they need to be connected multiple times. Of course, this is not applicable to humans in any way, because in mice, we connect them for 3 months, it's like 10% of their life. So in human, it's like connecting for 8 years, just not feasible. But the point, of course, here is that it's a fundamental science. So I think what it indicates that there's a potential to rejuvenate organisms. It doesn't mean that we reverse aging in a direct way. We just make them organism younger. Maybe some of the systems, they're still old, and some of them maybe age reverses very significantly, but overall, I think the organism becomes younger. And this is very interesting. For us, it becomes a gold standard. Now we relate this effect of parabiosis to many other interventions that we currently study. This study was published about a week ago in Nature Aging, so you can just go and look at for more details. Now, I'd like to mention another example of rejuvenation that we discovered recently, and this is a rejuvenation during embryogenesis. There are several papers that we published so far, but I would stress that so far it's only my lab, so it has not been validated by other labs, so we are looking forward to actually that somebody else would confirm the findings. What we find basically is that the biological age of the zygote, which is the fertilized egg, is not 0, slightly above 0. And then during early embryogenesis, it kind of initially stays the same and then suddenly drops to the lowest point, which we call ground zero. And it corresponds approximately to the stage of gastrulation. This is where we think aging begins of the organism. It's not when we increase mortality, not when we lose function, not at the conception, but we think at the gastrulation stage. So this corresponds to the lowest biological age so far. And we've done it using various omics approaches. Of course, we applied first Steve's clocks, but then this is an example of where we also developed a single-cell epigenetic clock. This was developed by Alex Trapp, a talented former member of my lab who now works at Retro. And he found that, for example, at the epiblast, you can see on the left, at a 4.5 days embryonic day, 5.5, 6.5, 7.5, there's a decreased age. The age is reduced. When we look at the different lineages, we find that all three germ layers are rejuvenated during embryogenesis. But interestingly, extra-embryonic tissues, like cells that would become placenta, they are not. So it does not equally apply to this system. So now we also recently developed a transcriptomic clock in addition to the epigenetic clock. And this clock works really, really well. This is the best works in mice, also mice and rats, but we also have mouse, rat, human clock. And this is based on over 3,000 samples that primarily we mostly sequenced. This is the many of the age-related changes, many in response to interventions. We sequenced almost the entire ITP set of samples. ITP is Intervention Testing Program funded by NIH. And transcriptomic clock, as you can see, works really well. I will just show you one example where we apply this to embryonic rejuvenation. And we again observe a reduction in biological age with the lowest point around gastrulation. So completely different omics approaches seem to indicate the same, lead to the same conclusion. But again, it needs to be validated by other labs. I would like to make another point, which is often overlooked in our research. And this is what actually age-related changes indicate. Often you go to a conference and you listen, somebody says, oh, this protein changes with age and decreased with age. That means it's really bad. Let's restore it back to the normal level. But I would argue that age-related changes tell us nothing, actually simply nothing about the causal inferences, whether we observe change or not. And this is illustrated by this example of the same man in the younger and older state. And you can see that this person, as he becomes older, begins to wear glasses and also uses hearing aid. These are age-related changes. This happens with age. And if we build a clock, these features end up in the clock. They can be used to quantify biological age. And of course, this is an example of just some external features, but at the molecular level, we observe the same thing. There are some molecular changes which happen during aging, they end up in biomarkers of aging. But they are not, I mean, in a way, they are not causal. In fact, they represent not just aging, but protection from aging, right? So the same way, molecular changes may happen that protect against aging, not being aging itself, but being part of the clock. And so Albert Ing, who is a graduate student in the lab, recently developed what we call causal biomarker of aging. And this is an epigenetic clock where he was able to carry a large genetic study to identify CpG sites which are causal to aging and also which are adaptive, and use them separately to build a clock. You can see on the top part called DAM-age. This is a clock based on damaging changes. It can be used perfectly to predict age of the organism. And the upper right, adapt age, which is a clock based on adaptive changes. These are responses to aging, but also can be used to quantify aging. And then on the lower right part, you can see the use of these clocks. When we apply clock based on damage, we can show that cells can be rejuvenated when we reprogram them to iPS cells. On the other hand, adapt age does not show that. What this means is that the clocks, are very useful. This is a really revolution in the field, but we still need to work and develop better clocks, clocks that are more enriched in kind of changes which reflect damage itself, and also other processes. So what I'm trying to say here is that we witness a revolution in the field with all these biomarkers when we can ask fundamental questions like when does aging begin, for example, whether we could rejuvenate organisms, how can we test interventions. This is amazing opportunities. But at the same time, there are still many challenges, and these challenges are not really addressed yet. For example, we don't know which biomarker we could use in clinical trials. So, of course, there are different opinions, but there's no consensus currently. So we just need more research, more funding, more effort, more talent that need to go to the field. So to quickly mention our effort in this, we recently formed what we call a biomarker of aging consortium. And this is a group of researchers, about 25 labs, leading labs in the field. And several are represented here, of course, Steve and Brian, Kennedy, and probably miss someone. And here is, this is led by Vadim Gladyshev, who is first author. He's a member of my lab sitting here. He will participate tomorrow in a panel discussion on biomarkers. But of course, many other labs. The first paper of this consortium will be published later this month in Cell. And we also would like to announce another conference, which is December 4th at the Bach Institute, where we would also, this conference devoted entirely to biomarkers of aging. Finally, this is a slide on a tool that we also developed called ClockBase. This was developed by Albert Ing, and this is what he did. He basically applied currently the best clocks, like, for example, PhenoAge or GrimAge and other clocks, Steve's clocks and some other clocks, to the entire GAO database. If somebody has done some research and plays GAO database, and you can go to this our tool, ClockBase, and simply identify the biological age, predicted biological age. We haven't even submitted a paper on this, but I think we wanted to make available for the community so that it advances research more rapidly. That's it. This is my acknowledgement slide, and people on the left, you can see it's members of my lab. I told you a little bit of the research done by Jesse, like on stress. This Alex Tishkovsky is longevity signatures and transcriptomic clock. Bohan Zeng, parabiosis. Madi is a consortium. And also Albert, who is a clock base, and also causal clocks, but several other people also contributed to this, as well as people shown on the lower part, which are former lab members. And of course, we also have many collaborators who contributed to all these studies are done in collaboration. We really like collaborations. And you can see several of these researchers here. It's amazing collaborators. And on the right, funding sources that supported this research. And I would like to thank you very much for your attention.